would like to thank Andreas for the invitation, and I would like to mention that my PhD was supported financially by Andreas, and also would like to thank my professor in UK, Professor George Metzgers. It is very well known that obesity rates are increasing rapidly, and this is because of two main reasons. One is that we increased our energy intake, and the other one is that we decreased our energy expenditure. And we accumulate excessive weight, mostly in the form of excessive fat mass. And this is okay, as long as we're healthy. But obesity causes health problems, and very serious health problems. And there are some recommendations from the World Health Organization. There are some interventions that indicate what we should do in terms of that. Mostly, we focus on diet and exercise to have very good nutritional habits, and also to increase our physical activity levels in order to have energy expenditure. However, it seems that these kind of interventions do not work, are not efficient. So we need to focus on something else. We need to redirect our research to assist the field. The interventions are okay, but we need to assist the field. And one potential field is to examine the fat cell. It is well known that we do have two kinds of fats in our body: the white fat cell and the brown fat cell. The white fat cell has huge lipid droplets and produces energy, stores energy and produces energy in the form of ATP. The brown fat cell is rich in mitochondria. It has small lipid droplets and produces heat to maintain body temperature. And while with a white fat cell we cannot spend energy because we need to increase our energy expenditure, with a brown fat cell it seems that we can, because heat is emitted to the environment. So if we spend energy to produce heat, we may lose weight. We may increase our energy expenditure. And this is something that increased the research in the area. You can see here how a brown cell works. The main difference in the lipolysis process is that within the mitochondrial matrix in the respiratory chain, when the protons goes out of the mitochondrial matrix in the intramembranous space, this is the time that ATP is synthesized. However, in a brown adipocyte inside the mitochondria, a unique protein is involved in the process. This is called uncoupling protein 1. This protein brings the proton inside the mitochondrial matrix and produces heat instead of ATP. So it bypasses the ATP synthase. This phenomenon is called non-shivering thermogenesis. And as I said before, this phenomenon can increase our energy expenditure. But how can we identify brown adipose tissue? How can we identify brown fat cells in our body? Many years ago, some physicians that they performed positron emission tomography and computed tomography. This is a technique, it is a scanner actually, that can identify cancer tumors. They inject glucose. The glucose is attached on metabolically active tissues, and they go with a scanner and they identify the cancer tumors. However, they noticed, some of the physicians, that there are some areas, some metabolically active tissues that are not cancer tumors. And they demonstrated that this was a brown adipose tissue, this was brown fat. As you can see here on picture A, this patient was exposed to cold for 2 hours prior to the examination of positron emission tomography. And you can see beyond the chest and along the spine area, those black spots. These are brown fat cells. And the same patient was not exposed to cold, and he undertook again positron emission tomography, and the only metabolically active tissue that you can see here is the brain and the heart. So brown fat are triggered by cold exposure, and we can identify them through positron emission tomography. But the thing is that we thought that till then, till 2009, actually, we thought that brown adipose tissue is not active in adults. We thought that only infants, they display the tissue, only infants, they have the tissue because after the delivery, they need to maintain life, so they need to produce heat. However, in 2009, two investigators, two groups actually, they used the technique of positron emission tomography and computed tomography, and they measured brown adipose tissue activity in healthy. And they also measured, one of the groups measured energy expenditure. And they found that there is a strong correlation between brown adipose tissue activity and energy expenditure. So, the more the brown adipose tissue activity in humans, the more the energy expenditure. And don't forget, this is in healthy. This was a major discovery. However, actually, the second study, sorry about this picture, the second study, as you can see here, they found the deposits through positron emission tomography and computed tomography in healthy again, but they undertook biopsies as well. So they wanted to confirm that this is the tissue. These biopsies identified, through the biopsies, they identified UCP1, the uncoupling protein 1. And also they identified the cytochrome C, which is an index of increased mitochondrial activity in the area. So these two studies confirmed that brown adipose tissue is present in adult humans. There were some evidence in animals, actually, from animal studies that indicated that if you do exercise, you may increase brown adipose tissue activity. But that was in animals. And our group thought that this is a very good idea. We may use positron emission tomography to identify the brown adipose tissue and measure physical activity levels at the same time. And we did exactly this. You can see here one of the pictures of the positron emission tomography that we performed in cancer patients. We made sure that this is brown adipose tissue and not cancer tumor. And we also measured at the same time physical activity levels with a simple way. We used a questionnaire and we performed some correlations to see if there is something in the area. That was the first step, a cross-sectional study. The results shown that the more the physical activity, the more the BAT activity in humans. Actually, it is in cancer patients. We had this limitation. However, the positron emission tomography in all possible corrections, body weight, body surface area, and lean body mass, this is brown adipose tissue, showed correlations positively correlated with physical activity levels. And also we found that the more the BMI, the less the BAT activity, which is something known, but for the first time, we split our sample, our participants in normal weight, overweight, and obese participants, and we found that through that categories that participants that they have increased BMI that are obese, they display the tissue, the brown adipose tissue activity less. And also we found some differences in sex between men and women, that this was very well known, and this is due to hormonal differences. The women display the tissue more than men. There were some, in 1980s actually, there were some investigators that they observed in some white fat cells in mice that UCP1 and uncoupling protein 1 was present in these white fat cells, and they couldn't explain why. So several investigators from 1980s to 2012, they were trying to figure out what is that the function of this protein, the function of uncoupling protein 1 within a white fat cell. And a group from Harvard in 2012, they proposed a new mechanism in terms of that. Now, we're not talking about the brown adipose tissue that it is located beyond the chest and along the spine area. We're talking about white adipose tissue that may display properties of brown adipose tissue. This tissue is called brown-like, or bent, or bright. This is a third kind of adipocyte in our body. This investigator said that when you do exercise, you trigger the action of a co-activator, the PGC1 alpha, within the skeletal muscle. That triggers the action of the FNDC5 gene. This produces the FNDC5 protein, all of that within the skeletal muscle. And this protein is then cleaved and released a product in the bloodstream that is a newly discovered protein. This newly discovered protein is called irisin. Irisin protein is then attached on the surface of white adipocytes, and through a cascade of events increases the expression of uncoupling protein 1 within the white adipocyte. And this is a very strong indicator that the mode of the white adipocyte changed to a brown-like mode. And they then suggested that this may increase our energy expenditure, and if we increase our energy expenditure the same as the brown fat cell, then we may lose weight and we may improve our insulin sensitivity, which is something very beneficial. And this directed them to search further about irisin. So they suggested that irisin may have a therapeutic role. And plenty of studies came up after this study to search about the role of that unique protein. However, some of the investigators, they put an argument in the area. This is a study from Timmins et al. That they examined actually the mRNA expression of the FNDC5 gene, which is the precursor of irisin, which is the gene that produces the protein, and then this protein is cleaved, and we do have irisin. And they did that in 500 people, and they performed some analysis, and they found that the FNDC5 mRNA, it is positively correlated with body mass index. This is exactly the opposite that we found in brown adipose tissue. It would be expected that FNDC5 gene to be inversely correlated with body mass index. And they did not find any correlation of the FNDC5 mRNA within the muscle with blood glucose and fasting insulin. They also said that FNDC5 gene is not equally expressed in humans. They found that to be expressed more in older than in younger individuals. In 2013, another study came up. An investigator examined the FNDC5 gene, and the group suggested that the FNDC5 gene in humans is completely different than in mice, than in gorilla, than in some other species. So they found that the FNDC5 gene displays a mutation and starts from a non-canonical start codon. So the protein that is produced, it is the product of a non-canonical start codon, which means that is the protein functional? Usually, if you don't produce a protein from a gene that is not canonical start codon, then the protein may not be functional, but this is not the case at the moment. They also performed some electrical stimulations of the muscle fibers, and they found that the FNDC5 gene is not expressed, is not triggered. So they put another argument, and this has to do with the fact that probably the possibilities to produce a functional protein are much more or less than we thought because of the non-canonical start codon. Many studies, again, investigators continued to put a lot of effort to search the area, and they suggested several pathways that irisin may have a therapeutic role, that irisin may benefit human health. The suggested pathways are, as I said before, through the white adipocyte. Irisin is attached in the white adipocyte, and through a cascade of events produces the protein irisin, the protein UCP1 inside the white adipocyte, causing a browning effect, and probably we may increase energy expenditure, we lose weight, we may fight obesity, so we can improve our insulin resistance. The other pathway is that irisin may affect osteoblasts, which means that we may decrease the risk for fracture. Another pathway is that irisin is positively correlated with BDNF. This is a protein that is expressed in brain, and it is responsible for cognitive performance. If it is increased, so we can improve our cognitive performance. And the last pathway was that irisin, it interacts with beta-trophin, which is a newly discovered protein as well, that may regenerate beta cells in our pancreas, which may help us to fight type 2 diabetes. All of that are promising. However, did they measure irisin? That's the question. In 2013, Atherton and Phillips, they said, okay, some studies came up. The most of the studies, they investigated the effects of exercise on irisin production, because irisin is exercise driven, so we should investigate that. Some very good studies, such as from Pekala et al., they said that we were not able to actually identify that irisin may cause a browning effect on white adipocytes. Even though they found that there is an effect of exercise on PGC1 alpha and FDC5, they could not determine that irisin may cause a browning effect, because UCP1 within the white adipocyte, even though it was increased, it did not display a significant increase. So Atherton and Phillips, they examined all these studies, and they said for the first time that probably this protein doesn't do anything. Probably this protein does not exist. That was the first time that someone questioned the protein. And suddenly, in 2015, Albert et al., they wanted to check this hypothesis. Is the protein functional? So what they did is they went back to see what are the methods that we're actually measuring irisin now. They measured irisin through Western blotting. Then some antibodies were developed in order to measure irisin through ELISA kits. And Albert et al. Actually, what they did is to check the existed methods that we measure irisin. And the first thing that came up, it was that the antibody that it was used in the initial phase to identify irisin, and this was in the initial phase from bostometrol, it was from a terminal that is not part of the irisin peptide. It was a terminal of the FDC5 protein that is not part of the irisin peptide. So an antibody was developed that was a polyclonal antibody, so probably this antibody attracts other proteins as well. It is not a unique antibody, so you cannot identify irisin. And also, this was confirmed by the buds that the irisin was detected. You can see here 32 KDA glycosylate, 24 KDA non-glycosylate, and in the validation study that Albert et al. Did, they found different values. And also for the first time, they used mass spectrometry, which nowadays is considered the gold standard to measure irisin. So mass spectrometry confirmed that irisin exists, but it exists at that kind of molecular weight in serum. And also Western blots techniques confirmed the same. So what they did is to examine all the available ELISA kit, not all of them, but the most important of them, the ELISA kit that they used the most for the irisin identification, to see if they actually measure irisin. And they determined that these ELISA kits and all these studies had been done so far, they don't measure irisin. They measure something else. And they went beyond that. They wanted to find out which was that protein that they actually measured. And they found that probably they measured apolipoprotein A1, which is the protein that could be identified with Western blots and that kind of bands. So that changed the world, that changed the whole research around the area. Our group initially wanted to test the hypothesis, the mechanism from the muscle from the exercise stimuli to the muscle, to the circulation, to the white adipocytes. So the first thing we did is to conduct a systematic review. This is a very strong design because if you are able to accumulate evidence from several studies, then you may be able to determine a conclusion, to form a conclusion. So our aim in this systematic review initially was to systematically identify evidence of the effects of physical activity / exercise on the link between PGC1-alpha, which is the co-activator that triggers the FNDC5 gene, and FNDC5 in muscle, circulating irisin, and UCP1, which is the direct index that can determine the Brownian effect of white adipocytes, of white adipose tissue in humans. And we did that only in humans because we wanted to see what is the value of these studies, the existing studies. We used an appropriate algorithm. We used two databases, PubMed and Embase. We retrieved pretty much 2,000 articles, and we included in our analysis 45 articles. We used the PRISMA guidelines. These are specific guidelines how to perform a systematic review in order to retrieve accurate and valid results. We evaluated our studies. The first thing we did is to evaluate the risk of bias. And this was through a tool from the Cochrane library. We split our studies in randomized controlled trials, single group design studies, and cross-sectional trials, and cross-sectional studies. As you can see here, the red color is high risk of bias. The green one is low risk of bias, and the yellow one is unclear risk of bias. So in these questions here, there was a lot of risk of bias of the existing studies. The other thing we did is to evaluate the quality of the reporting of the results. Sometimes when you write a paper, you may hide an information. So we evaluated that. We used tools, again, from the Cochrane library to identify if an information is hidden in every paper, in each paper. And as you can see here, with a consort tool, we evaluated the randomized controlled trials, the controlled trials, and single group design studies. And overall, the half of the studies, they did not display the essential information. And the same, 40% of the studies, it is in cross-sectional studies. We evaluated this with a different tool. This is the STROM tool. Again, this is not the quality of the study, but the quality of the reporting of the results that sometimes helps you to form a conclusion in terms of the results of the studies. Overall, we found little evidence to determine the link between PGC1-alpha mRNA and FNDC5 mRNA in human muscle in response to physical activity. We were not able to determine any conclusion in terms of that. 45 studies, we couldn't find any link between PGC1-alpha and FNDC5. There were limited evidence in terms of uncoupling protein 1 within the white adipocyte. Actually, we found only one study, so we cannot form any conclusion. And the other thing we did is to assess the methods that irisin was measured in these studies. And we cannot form any conclusion again because the methods were inaccurate. However, we identified two papers, two articles that they used mass spectrometry, and these articles were subsequent articles of the initial study from Albert et al., and they used exercise to measure irisin through mass spectrometry, which is considered the gold standard. However, these studies display several limitations. The one study had only post hoc measurements, so even though they had a control group, they did not measure before and after the exercise program. They just measured after the exercise program. And the other study, it was a single group design study without a control situation. So again, we cannot say that exercise increases irisin in the circulation. It is needed further research here. One finding of this systematic review is that we identified that irisin is present in human circulation. However, this is 10 to 100 folds lower than commercial ELISA kits. And this shows the difference. Actually, it confirms the finding from Albert et al. That the irisin peptide is not the molecular weight of the peptide is not so big as they used to report. This was the one part. The second part was a randomized control trial. So we wanted to test part of the mechanism. And one thing that we did is to test UCP1 and coupling protein 1 within the white adipocyte after the exercise stimuli. And also we included some other indicators that can give us an indication if there is a Brownian effect here. If you identify UCP1, you can directly determine that there is a Brownian effect. But PGC1 alpha is an indicator for mitochondrial biogenesis by the external stimuli. PPR alpha, it is a factor that it is involved in the process within the white adipocyte to increase UCP1. And PPR gamma, it is present in brown adipose tissue and it is involved in the differentiation of the cell. So according to the literature, we thought that it would be a good idea to test that as well, because some investigators, they proposed that in the past. And our aim was to examine the effects of different types of exercise and detraining on these genes. Actually, we measured mRNA expression of subcutaneous white adipose tissue in healthy and trained men in a parallel randomized controlled trial. We interviewed 183 people. And we included 32 people, 32 individuals in our study. We had 4 groups, 3 exercise groups, and 1 control group, an aerobic exercise, a resistance exercise, a combined exercise group, resistance and aerobic, and of course a control group. You can see on the bottom the individuals that included in the final analysis after the dropouts. This is the experimental protocol. This was something that we were discussing with Andreas before we started the experiment. We were about what we're going to do. We're going to measure at baseline and after the exercise program, and how much would be that, how many months, how many weeks, and what kind of program we're going to use. But we thought it would be a good idea to include a detraining period. This is a methodology that it is used in clinical trials. You measure, you give the medicine, you measure again, you take the medicine and you give a placebo, the patient don't know about, and you measure after a while to see if the medicine works. So that was our idea in terms of that kind of design. So we took measurements at baseline, at 4th week, at 8th week after the exercise period, and also at 16th week after the detraining period. And we performed subcutaneous white adipose tissue biopsies. So we took biopsies from the belly through a surgical technique, body composition measurements, diet recall, physical activity. We measured maximal oxygen consumption and 1 repetition maximum to see if the exercise program was effective, and several anthropometry indices. We did not find anything. We detected no significant differences between and within groups in delta scores of the mRNA expression of UCP1. This is something that we use sometimes in statistics. We subtract one time point for one mean from another mean because we don't have a normal distribution. It is acceptable to do that in order to be able to perform an ANOVA analysis afterwards. So we found no differences even with the exercise stimuli or the detraining stimuli. However, when you do a randomized control trial, you include some additional statistical analysis because you want to see the mechanistic value beyond your findings. So we included in our analysis another statistical analysis that's called Cohen's d effect size analysis. And this is something that you subtract the mean values and you divided them by the average of standard deviations. And you get a number. That number would be between, in this case, because there are some other effect size analysis, in this case, could be between 0.3 and 0.4, which is a small effect, 0.4 and 0.7, which is a moderate effect. If you find something here, it means that you need to explore further. You need to investigate further. If you find something here, which is a large effect size, a value more than 0.7, then probably there is a significant difference there, but it is not detectable by the statistical analysis due to the form of the statistical analysis. And we found some very interesting indications, because this we cannot form a firm conclusion through the effect size analysis, but we found some indications. We found some that aerobic exercise increased UCP1. This is a UCP1 mRNA. From baseline to post-exercise, this is a small effect size. And from post-exercise to detraining, we found in the combined exercise group, which is resistance and aerobic, again, a moderate effect size, which is, again, the UCP1 is increasing. This is very interesting. That's why I was asking before the Sergei in terms of the delay effect. And we found that in controlled group, which is not normal. There are several explanations in terms of that, but we're continuing our analysis in some other factors in order to determine why is that happening. The same in the PGC1 alpha. This is only in post-exercise and detraining values. So PGC1 alpha declined in all the exercise groups. And in some of the cases, we found a large effect size. If we perform, for instance, a t-test here, we will find significant statistical differences. And I did that, and there is a significant difference here. In PPR alpha, the same. But again, we found in control group a decline after the exercise, after the detraining period. And in PPR gamma, we found in aerobic exercise group between post-exercise and detraining a large effect size, so PPR gamma was decreased here. And in the combined exercise group, we found that PPR gamma was increased after the exercise program. And in the resistance exercise group, we found an increase after the exercise program, a decrease after the detraining period in PPAR gamma. But one very interesting thing that we found in this study is that we detected at baseline UCP1 positive cells, white fat cells. And we need to see why is that happening, why we were able to identify UCP1 within a white adipocyte without any stimuli. This is a baseline values. These are baseline values in untrained individuals, of course, in a variety of body mass index and age. So probably it needs a careful exploration. But this is very interesting, very interesting. Our conclusion is that different types of exercise in the subsequent detraining periods have no effect on the mRNA expression of UCP1, PGC1 alpha, PPR alpha and PPR gamma genes of white adipocytes in healthy untrained men. Nevertheless, there is a trend of increasing UCP1 values after the aerobic exercise program that is also appeared in the detraining period in the combined exercise program. Overall, to answer the initial question, can brown and brown-like adipose tissue benefit human health? We now know that increased brown adipose tissue activity may increase resting energy expenditure in humans that may benefit health. We know that brown adipose tissue activity in humans is increased by cold exposure. However, we don't know if it is increased by the exercise stimuli. Further research is needed to explore whether physical activity exercise affects brown adipose tissue activity. There is a huge project ongoing in Spain nowadays that is doing exactly this. They will examine the effects of exercise on brown adipose tissue using on classical brown adipose tissue activity using positron emission tomography. There is evidence that brown-like adipose tissue is expressed in humans. However, further research is needed to explore whether physical activity/exercise transforms white into brown-like adipose tissue, especially when we examining irising. So it needs further research in the area. And with this, thank you very much for your attention.